drug combinations. Let's get into it, because they're not just, an alternative strategy. They're really the backbone of how we fight our toughest diseases.
Yeah.
We're talking about everything from cancer to superbugs. And the goal isn't just adding one drug's effect to another. We're hunting for synergy.
Right, that's the keyword, synergy. It's that moment where 1 + 1 equals, say, 10. It's an effect that just completely blows past what you'd expect from the individual parts, beyond what a model like Bliss Independence would even predict.
And the search for that synergy is, well, it's been incredibly slow.
Glacial is the word. And the reason why is a fascinating math problem, really. The complexity just explodes. It scales up quadratically with every new drug you add.
Quadratically.
Yeah. So, testing 10 drugs one by one, simple. But testing every possible pair from a group of 100 drugs, you're suddenly looking at nearly 5,000 experiments. It gets out of hand so fast.
And that forces researchers into a really tough spot.
A terrible trade-off. Do you test a huge number of combinations but only scratch the surface? Or do you test just a few, but really, really deeply across many different doses?
And you need that dose density to find the real patterns, right?
You absolutely do. Otherwise, you might miss everything.
Well, breaking through that exact limitation is what our deep dive is all about today. We've been looking at some amazing source material on how technology is finally helping us escape that trap. We're going to unpack two big frameworks. First up is Combocat, which uses robotics and machine learning to just massively increase the throughput.
The sheer number of tests you can run.
Exactly. And then we'll look at a system called Express, which takes all that data and uses explainable AI to give us real biological understanding. Okay, so let's start with Combocat and this throughput problem. You mentioned that trade-off. Traditionally, drug screens were stuck with these small kind of sparse formats, maybe a 3x3 or 5x5 dose matrix.
Yeah, and that's just not enough. You're basically looking at the drug landscape through a keyhole. It's so easy to miss something important.
So how does Combocat get around that?
Well, it's a really smart integration of hardware and an open source workflow. But the absolute key piece of technology is the acoustic liquid handling system.
Acoustic liquid handling. That sounds like science fiction.
It almost is. You have to picture it. It's a robot that uses sound waves to move liquid.
Sound waves.
Yep. It dispenses these tiny nanoliter volume droplets of drugs. And because it's using sound, there's zero physical contact. No pipette tips touching the solutions.
Wow. So resource use goes way down, and you basically eliminate cross-contamination.
Precisely. And that speed and precision is what enables Combocat's first mode of operation, what they call dense mode. This is all about getting the highest quality data possible. It measures every single pairwise dose combination in a big, comprehensive 10x10 matrix.
So 100 measurements for every single drug pair.
100 measurements, and they run it in triplicate to make sure the data is rock solid. It's like taking a high-resolution photo of the entire dose-response landscape.
So how big of a picture did they end up with?
Using this dense mode, they built this incredible reference data set. 806 unique drug combinations, which added up to over 290,000 individual measurements.
That's a serious benchmark.
It is. It became the gold standard data they used to train all their models later on.
But first they had to prove the system even worked, I assume. Did they, replicate some known synergies?
Oh, absolutely. That validation step is critical. You have to build trust in the platform. They tested some classic combinations, for instance, sulfamethoxazole and trimethoprim-SMX and TMP, which is a standard antibiotic combo used against things like E. coli.
Yeah.
Perfect synergy, just as expected. They also confirmed it in cancer cells with a combo of AZD3-90 and CX-5461 in neuroblastoma, which is already in the literature.
So the foundation is trustworthy. Dense mode works.
Exactly. It provides a high-quality, reliable foundation.
But 806 combinations, that's an amazing data set, but it's still just a tiny fraction of what's possible. That dense mode sounds way too resource-intensive to scale up to, say, tens of thousands of combinations.
And that is the perfect transition. That's exactly why the machine learning is so necessary. And it brings us to the real game changer here, sparse mode. This is where they use predictive modeling, trained on all that great dense mode data, to get to ultra-high throughput with just a fraction of the resources.
Okay, break down how sparse mode works.
All right, so the genius is in the setup. First, they miniaturize everything even more, moving to a 1536-well plate format. Then, and this is the critical part, they cut the number of measurements by 90%.
90%.
90%. Instead of measuring all 100 points in that 10x10 grid, they only measure the 10 points along the diagonal where the drugs are at a 1-to-1 ratio, plus the responses to each single drug. That leaves 90 spots in the matrix completely blank.
Okay, wait a minute. Predicting 90% of your data sounds incredibly aggressive. How do you not just end up with a bunch of noise or false positives?
That is the critical question. And it all comes down to how good your prediction model is. They use what's called an ensemble model. They actually have 90 separate dedicated regression models, one for every single blank spot in that grid.
So it's highly specific.
Very specific, and it's trained on that amazing dense mode data, which is super high quality.
So the big question, how accurate was it?
The accuracy was phenomenal. The model hit a median R-squared of 0.945.
What does that mean in simple terms?
It means the model is successfully explaining and predicting almost 95% of the variation in the cell response. The correlation between what they predicted and what they actually observed was 0.98.
That is incredibly high confidence.
It's exceptional, and the reason it works so well is that it's a proximity-based system. The prediction for any given blank spot relies most heavily on the real measured data points right next to it.
So it's not just guessing wildly, it's making very educated interpolations.
Exactly. It makes the results robust and biologically meaningful.
So what's the payoff? If this ML shortcut works, what kind of scale did it unlock for them?
This is the amazing part. Using sparse mode, they screened 9,045 unique drug combinations in a single neuroblastoma cell line.
9,000.
9,000. The researchers themselves describe it as the largest dense combination screen ever reported in a single cell line. And they did it with the resource footprint of a sparse screen.
That is a genuine leap forward. Did they find anything important in that massive screen?
They did. And it was a beautiful confirmation of biological principles. The strongest synergies they found were consistently in combinations that targeted complementary DNA damage repair pathways.
Okay, so that makes sense. DNA damage repair is like a cell has a couple of different backup power generators.
That's a perfect analogy. The two pathways here were ATM and PRRP. They're separate but crucial repair systems. So they hit the ATM pathway with one drug, AZD1390. That forces the cancer cell to rely completely on the PRRP pathway.
And then you hit it with a PRP inhibitor, like olaparib.
Exactly. You add olaparib or rucaparib, and you shut down that second backup generator. The cell's genome collapses, and it triggers self-destruction. It's a classic case of synthetic lethality.
So Combocat gives us the scale to find these things systematically, not just by guessing. But this leads us right into that classic problem with machine learning, the black box. The model can tell you that a combination works with 95% certainty, but it can't tell you why it works.
And that is the critical pivot. For a doctor, for a scientist, because the algorithm said so, is just not a good enough answer, especially when you're dealing with incredibly complex, high-dimensional data like a patient's entire transcriptome, the activity of thousands of genes all at once.
So you need a new tool.
You do. And that brings us to the second framework, Express. It stands for Explainable Predictions for Gene Expression Data. And this was designed specifically to build models that are not only accurate, but are also crucially biologically interpretable.
And they applied this to acute myeloid leukemia, or AML, a very tough cancer.
For a very tough, very complex cancer.
So how does Express crack open that black box? How do you make a complex model show its work?
It's really a two-pronged approach. First, they use ensembles of models. For this specific AML data, they found that a type of model called XGBoost worked best. And second, they pair these models with feature attribution methods. The main one they use is called Shapley values.
Okay, Shapley values. Can you break that down for us? In plain English, what is it actually doing?
Sure. Think of it like a team project. At the end, you want to figure out who contributed what. Shapley values are a method from game theory that does just that. It looks at the model's final prediction, say high synergy, and it assigns a specific amount of credit to every single input feature.
So every gene, every drug.
Every single one. It will tell you this prediction was driven 50% by drug A, 20% by the high expression of gene X, and so on. It turns a black box answer into a detailed, weighted explanation.
And using an ensemble, a group of models, makes that explanation more reliable.
Exactly right. By training lots of models and averaging their explanations, you filter out the noise. Any spurious correlations, things the model latched onto by accident, they tend to just cancel each other out, leaving the real biological signal much clearer.
So what was the data they fed into this?
It was a huge data set. They had gene expression profiles from 285 real AML patients, and they paired that with synergy measurements for 133 different drug combinations, which gave them over 12,000 samples to analyze.
Okay, a ton of patient data. After all that analysis, What was the single biggest biological signature that the AI flagged as being predictive of synergy?
The major finding, the big global pattern, was the discovery of a very strong hematopoietic differentiation signature.
Differentiation. So how mature the cancer cells are.
Exactly. The model basically said that the most important factor for predicting synergy wasn't just which drugs you used, but the developmental state of the leukemia cells themselves.
And it pointed to specific genes driving that.
Oh, yeah. For instance, high expression of a gene called MYES1 was strongly correlated with higher predicted synergy. And IS1 is a known bad actor in some aggressive forms of AML. It's linked to a certain cell type that's often resistant to therapy.
Wait, so the AI is pointing to the cells we think of as the most resistant.
And it's saying they're actually the most vulnerable to the right combination therapy.
That's fascinating.
It is. And conversely, a high expression of another gene, DLL3, which is linked to worse survival, correlated with lower synergy. The top gene linked to positive synergy was CIDED2, which is critical for maintaining hematopoietic stem cells. The model was zeroing in on this whole axis of cell maturity as the master switch.
Which means you can start tailoring therapies. Did the analysis show that some drugs worked better against the stem-like cells versus the more mature differentiated cells?
It did, absolutely. Express split the drug combinations into two clear groups. There were combos that were more synergistic against the more stem-like tumors, and others that worked better on the more differentiated tumors.
Give us the key example.
The key example involves a really important AML drug called venetoclax. The AI found that combinations with venetoclax were significantly more effective when the cancer cells had a more differentiated or monocytic signature.
I just want to pause on that because that is a huge insight. We know those monocytic cells can be really resistant to venetoclax on its own.
That's right.
So the AI didn't just find a correlation. It identified the exact profile of a resistant cell and then pointed to the specific partner drugs that could overcome that resistance.
That is the power of explainable AI. The output isn't just a prediction score, it's a testable clinical hypothesis. It tells you how to go after the resistant cells.
And it got even more specific than that. You mentioned it could zoom in on the mechanisms of individual drugs.
It can. By using something called the Shapley interaction index, they can measure how much a gene's expression influences a specific drug's effectiveness within a combination. So for venetoclax, the interactions pointed, unsurprisingly, to genes involved in apoptosis-programmed cell death.
Its main job.
Its main job. But what's really cool is that it found increased expression of BCL2, the drug's target, was linked to increased synergy. This confirms the idea that while those cells are vulnerable, you need that second drug to block their escape routes.
And did it work for older drugs too? What about a classic chemo agent like cetarabine?
Yes, and this was a fantastic validation. For cetarabine, the AI immediately highlighted genes involved in the metabolism of organonitrogen compounds. It specifically called out two genes, CDA and NT5C2.
How are those important?
They are the two major known genetic factors that determine how a patient responds to cetarabine therapy. Express acted like an unbiased detective and rediscovered decades of medical research just from the data. So to sum it up, what we have here are these two incredibly powerful complementary frameworks. Combocat gives you the scale. It lets you test thousands of combinations with a level of detail we just couldn't get before. And then Express gives you the understanding. It takes that mountain of data and turns it into real biological insight, like confirming just how critical a cancer cell's differentiation state is for synergy.
And what's really exciting is that these tools aren't being locked away. The researchers have made them open source. The Combocat protocols, the code, even those huge reference data sets are available for other scientists to use and build on.
And that open source philosophy really sets up the final provocative thought here. Imagine if this becomes a community effort. If research groups all over the world start contributing their own high-quality, dense combination data.
A global shared data set.
Exactly. These data sets could be used to constantly retrain and improve the machine learning models at the heart of systems like sparse mode and Express. It would create this dynamic, living resource that gets smarter and more accurate over time for the benefit of everyone. So the final question is, what complex, lineage-specific vulnerabilities might thousands of new crowdsourced synergy maps reveal when they're built not by one lab, but by the collective power of global data? We may have just seen the beginning of a truly collaborative, AI-driven future for drug discovery.